I want to thank the organizers, especially Jim, for inviting me here. That's really a great honor for me to speak among my heroes, essentially. I hope to tell you at least a couple of things that are a little bit new. It's always a bad idea to do data analysis on the day of the talk, but at least you're not bored by what you're talking about because you've said it many times before. So since this is supposed to be very interactive, please interrupt me as much as possible, not just in the 20 minutes at the end, but even before that. Okay, my title is Representational Similarity Analysis of Visual Object Population Codes. And I want to do two things today, talk about my general research program and about two recent studies. So in terms of the general approach, the classical approach to fMRI is activation analysis, where we look at sort of the low spatial scale of activation with an imaging technique, often smoothing the data, and we consider each region basically a unitary structure for which we only have a univariate estimate of the overall activation. And the general rationale for this is that activation indicates the involvement in some cognitive function of a region of interest. And more recently, inspired in imaging to a large extent by Jim's work, people have been interested in pattern information analysis, where we're sort of zooming in on a part of the brain and then looking at the pattern within a region and analyze that for the information it contains about the stimulus, typically. So the rationale of this approach is that the information in those patterns indicates the representational content, right? And of course, that's an interpretation and somewhat problematic, perhaps, but that is the motivation for taking this approach. And this motivates using high-resolution imaging and multivariate analysis on these patterns of activity. To me, this is exciting, and to me, this is the place where the action is currently, because it kind of promises to fill the explanatory gap between cell recording and functional imaging. So within, at this intermediate level of description, at this intermediate spatial scale, you might say, between the single cell level and the level of the functional region, there have been these pattern information studies, and not just in functional imaging, but also in cell recording. And I think there's sort of a greater integration in our communities emerging through this approach to some extent. So we can look at these levels of description also in terms of theory and experiment. And then on the side of the experiment, we have behavioral measures at the largest level of description, at the largest scale. Standard fMRI, high-resolution fMRI, and neuronal recordings at the lowest level. On the side of theory, we have the box and arrow models of cognitive psychology at the top. Then we have cognitive computational models, which are not neuronal in that they're not, so they don't have lots of units and do parallel processing on those units, but they're fully explicit computational models. And then we have neural network models and single neuron computational models. So historically, different disciplines that have been interested in the questions that we are interested in have made the link between theory and experiment at different levels here. For example, cognitive psychology made this link at the top level between the box and arrow models and behavioral measures. And all the way at the bottom, computational neuroscience has made the link between single neuron computational models and neuronal recordings. Cognitive science was the attempt to make the link between behavioral measures, as in cognitive psychology, to cognitive computational models. And more in the last 20 years, perhaps, has been somewhat overshadowed by cognitive neuroscience, which in a sense, went back to the box and arrow models, just taking a step back, if you will, and linked that up with standard fMRI, sort of drawing the connections between the boxes and the brain blobs. So what's really lacking is the connection between experiment and theory at this intermediate level of description. And that's sort of the level that I'm focusing on in my research. So we want to connect these cognitive level computational models and neural network models to standard and high resolution fMRI, and also to neuronal recordings and behavioral measures, of course. The single neuron computational models, I think, will not ultimately explain cognition. So at the level that I'm interested in brain function, they are not going to be the final solution. And the box and arrow models, they're not completely specified. So they're sort of just sketches for models, I would say. So the core concept of the approach that we're taking to this is the concept of the representational dissimilarity matrix. So what is that? We present a number of stimuli, and for each stimulus presented to the brain, we estimate a response pattern in a given region of interest. And then we compare these response patterns, and we compute a dissimilarity, typically as 1 minus the correlation over space. But it's interesting also to explore other distance measures for that. And so this gives us a matrix, because the matrix tells us this dissimilarity for each pair of the stimuli. So it's indexed vertically and horizontally by the stimuli, and symmetric about a diagonal of zeros, typically. And this matrix essentially addresses the question which distinctions between stimuli are reflected and which are lost in the population code. So that goes back to some of the issues that came up yesterday. For example, when you're interested in metamares, you're interested in them because these are two stimuli that are not distinguished by the code. So that tells you something about the code. But equally interesting, of course, is what distinctions are emphasized by a code, right? So a weaker way to state this is what distinctions are emphasized, what distinctions are de-emphasized. And the de-emphasized distinctions in functional interpretation would be that these are irrelevant dimensions or accidental properties to be abstracted from. So when we do take this approach for a set of images like this one, we get a matrix like this. So this is quite a rich matrix because it has about 100 images along the 2 dimensions. So what does this look like? Here I'm using this color scale with warm colors indicating large dissimilarities, so clear distinctions. And this is what it looks like for a human IT measured with fMRI with 2 millimeter voxels. So as you can see here, there are some things that are immediately obvious. When one of the images is animate, the other is inanimate, then the dissimilarity tends to be large. When both images are human faces, then the dissimilarity tends to be very small, right? But more importantly, we have this very rich pattern with thousands of entries that can serve as a kind of signature of the representation, right? And of course, that's limited by our stimulus set, but that's true of any analysis, right? So it's an interesting and rich characterization about what the region cares about and what it might abstract from. And importantly also, despite its being so rich, it does summarize the information across the response channels, right? Because each dissimilarity is the dissimilarity between 2 patterns, so it kind of combines the evidence across voxels, and that is useful because it utilizes the major strength of fMRI as compared to cell recording, for example, which is the large number of channels. But it's also interesting for cell recording, especially when we have a lot of different neurons, either sequentially measured or simultaneously measured. Is there a question?
I was just looking at the percentile scale, and I'm just wondering what range of actual correlations the like 0 to 100 range on the percentile scale corresponds to.
Oh yeah, I have that figure. I don't have it here. Maybe I'll show it to you afterwards. So here it's all rank ordered, so all the colors are equally frequent, and that emphasizes the distinctions. That makes it easier to see the distinctions. We have the full distribution, but so it's on Marika's poster. I don't know, is that still up?
Yes, it is.
So all this work was done together with Marika Moore, sitting over there. Maybe I'll show it to you afterwards. It's quite a broad distribution. So we can also use the signature to compare representations in different species. For example, here's the human IT representation of those images and the representation of the same images in the monkey. And now, because these matrices are indexed by the stimuli, they're very easy to compare, and we're obviating the need for defining the correspondency between the places in the brain in the monkey and the human. And in this case, the monkey IT was measured with cell recordings, actually. So this is sequential cell recordings from 674 cells in monkey AIT to the same images. So it's even more striking that the similarity is really quite large when you just look at the matrices. But the sort of same categorical divisions appearing to explain a lot of variance here. And we've done sort of detailed analyses also on the correlations within the categories, and many of them are also highly significant. So another approach we can take to this kind of data is to add an assumption, which is the assumption that there might be a categorical structure inherent to the representation. Now we can add that assumption without assuming a particular category structure, so it's still sort of more data-driven than the classical object vision fMRI approach, where the categories are predefined and the different responses to different images within a category are averaged across. And when we do this here, we get these clusters for faces and bodies and for inanimate objects. And this is very similar to what we get for the monkey. And for the monkey, this has been explored in much greater depth and also for a larger set of images, over 1,000 images, in Rusbe-Chiani's paper in the Journal of Neurophysiology. So another thing that we can do using this signature of the representational dissimilarity matrix is characterize model representations with it. So for this purpose, we just present our stimuli to computational models, and then we get these activity patterns in the models, and just as we did for the brain. And so one approach for testing the models would be to directly compare these model activity patterns to the brain patterns. But again, just as for the comparison between human and monkey, we face this problem of needing to align the patterns. And Jim has talked about this in the morning session. So that's really a challenging problem. One approach to it is the approach that you've heard about in the previous talk, which is voxel receptive field modeling, where you use a linear model to predict the voxel responses here from the internal responses of the computational model. But what we can also do is go to this slightly more abstract level of the dissimilarity matrix and compare the representations at this level. And this is attractive because it obviates the need for defining this correspondency. So we don't need to fit this linear model at this stage, and we also don't need to control for overfitting that is associated just with establishing this relationship. Right? We can just directly correlate these dissimilarity matrices. And that's what I call representational similarity analysis. So when we look at a few model dissimilarity matrices for these 92 images, this is the matrix for the H-max model. This is the matrix for a V1 model. It's the one that's part of the H-max model, so that's the simple and complex cells of that one. And this is just correlating the RGB stimulus images. So what you can see there is that we see structure, but we don't see the kind of categorical structure that was immediately apparent in the IT matrices from both human and monkey. So when we quantify that a little, we can compare different models. And what I've done here is I've plotted the deviation from the human IT dissimilarity matrix as 1 minus the Spearman correlation. So I'm just correlating every model matrix to the human IT matrix and then doing 1 minus so that it's a distance. So the best one is the one with the smallest bar, and they're ordered by the size of the bar. And now these matrices come from different sources. The ones that are labeled in red come from other brain regions, because we can use this to compare brain regions as well. The ones labeled in black are sort of naive models as sort of simple image transformations. The blue ones are neuroscientifically motivated computational models. And the green ones are simple category models that don't really compute anything. We're just assuming a category structure, and we're assuming that we have some mechanism to identify the category. And so this exercise on this data set gives a little bit depressing results. The best fitting model is the animate-inanimate model that just emphasizes this distinction. So it just has ones wherever you compare an animate to an inanimate and zeros everywhere else. Then the most similar other models here are other brain regions, FFA and early visual cortex as well, shares a component with the IT. And the model that we want to fit well, which is in this case the H-max model, which is the only sort of higher level IT level computational model included, didn't really work very well to explain the dissimilarity variance here. So our job is to take these kinds of models developed in computational vision and in computational neuroscience, like the model of Tommy and Thomas Serre. And then, and this is, you've seen this before, this is sort of one of the few models that's really very closely motivated by the neuroscientific literature. So it takes a lot of information from the literature about these brain regions, and all the stages of the models are labeled according to the brain region that they refer to. But nevertheless, it's not directly constrained by brain activity data, right? So I think an important step is to try to bring these computational models closer to the data, in the sense that we use them in data analysis and we use them to predict the dissimilarity structure. So our job is to characterize each stage of models like this one by a dissimilarity matrix, and this is just a cartoon slide here, and then link up these model representations with the brain representations, right? And so far, sort of initial evidence, and this is sort of talking more about the future direction for the next 5 years or so, initial evidence suggests that the stage of category distinction, which in this model is associated with prefrontal cortex, looks most similar to what we see in LOC in the human, right? And this raises sort of the interesting question what AIT in the human is doing and what PFC in the human is doing. So I'd like to convince you that by using this level of description of the dissimilarity matrix, we can make these links at this crucial intermediate level of description. And what's very important to me also is to take both a hypothesis and a data-driven approach to analysis and learning from empirical data. So there's this continuum between data-driven and hypothesis-driven, and most researchers have kind of a preference somewhere along that continuum. And that's very interesting to me. So what motivates these preferences? There are positive and negative aspects to any point on this continuum, right? So there's no answer to this, that either data-driven is better or hypothesis-driven is better. We clearly need both. The good thing about data-driven analysis is its broad sensitivity. So we have the chance to find unexpected strong effects, right? And what could be greater than finding effects that are both strong and unexpected? You gain a lot of information, right? So this is sort of a very strong motivation for doing this. The upside of hypothesis-driven approaches is that we have very focused sensitivity. So we can find more expected weak effects that might be important for distinguishing theories, right? Of course, they can't, if they were totally expected, if we were already certain what we find, then we don't learn anything, right? But we're playing this game of saying, okay, we're reducing the possibilities to a very small number and then using the data to adjudicate between these. And that means this focus is sort of like the data are like the light, and we're focusing the light on a very small spot, and then we can see very clearly at that spot, but we might miss the big picture. So this brings us to the downsides of both of these. The data-driven approach can be a fishing expedition and not optimally sensitive to weak effects that distinguish theories that are of interest to us, right? So we might get a lot of information, but that's information that's not really relevant to the theories we're interested in. And the hypothesis-driven approach has the drawback that it's usually assumption-laden, and this has two aspects to it. One is that the results are only valid if the assumptions hold, right? And often these assumptions are implicit, and I think this is really a big thing that we need to reflect on much more, that in many studies where sort of we accept the analysis as valid, there's actually a lot of assumptions that are very questionable and that everything rests on. And the other drawback is that this assumption-ladenness leads to ignoring major variance explaining factors, right? So by focusing the light on a little spot, you might see something, and then you make a big story out of something very small when there's a big elephant in the room that you haven't seen at all, even though it would explain a lot of your data and might ultimately be more interesting. So how does that relate to our approach of using representational dissimilarity structure to test models? Well, a hypothesis-driven approach might be to distinguish two models. So for example, we could go to the literature and have two competing models for a given brain representation. And we can think of these models as points. And since we're characterizing them by dissimilarity matrices, these would be points in dissimilarity matrix space, right? So we'd have the V1 model would be a point, and the Hmax model would be another point. Now we could say, okay, we just focus on this one dimension. We're taking a very hypothesis-driven approach. And then we get the data, for example, from the early visual confluence for central stimuli. And we see that it's closer to one than to the other, and we do some statistics on it and draw some conclusions. However, the problem of this is that when we remove this assumption that of just looking at this one dimension of the model space, in this case we see something like this, that actually our early visual dissimilarity matrix is about equally far away from both of these models, right? So the major thing here is that both of these models don't work. And if we allow more models, then we're sort of in this more complex picture where everything it goes sort of continually as you add models or parameters from hypothesis-driven to data-driven, right? So now you're looking at sort of a larger number of models, and you see the bigger picture, and that's confusing. On the other hand, once you've looked at that, it's very hard to go back and just choose a small subset of this and then feel like you can be confident about any strong conclusion. So in a way, it's bracing, but it's the kind of thing that I think we need to look at. So here I've used the same color code as before, and the major thing that I take from this, so this would make it a data and hypothesis-driven approach now because there's so many models. The major challenge that this suggests is that all the brain regions are sort of in a cluster, and all the models are sort of around, but they're not really invading that cluster and allowing us to match up one-to-one which model explains the representation in which brain region, right? So that I view that as sort of the challenge for the next 5 years, basically, to explore more models and to also change the models, tweak the models, or build new models that do better at explaining our actual data. So our conclusions from the prior study that I showed you some highlights of were as follows. I'm summarizing them because the two more recent things that I want to talk about build on that. So we saw matching categorical representations in man and monkey, and we found the same major category clusters in both species. Animate and inanimate was the major division, and face-body was a subdivision among the animates. We also saw, and I haven't shown you this, a similar continuous within category representation. So within categories, IT seems to place the visual objects in a very similar continuous object space. And from the computational model tests, we saw that the categorical clustering was absent in models using either low-level features or high-level natural image features. And our sort of working hypothesis is that IT may employ features that are optimized for distinguishing conventional categories. So that would suggest that it's not enough to do unsupervised learning and just represent natural shapes, but that the behaviorally relevant divisions and something like animate and inanimate would seem to be behaviorally relevant to all primates equally, might inform the selection of those basis functions in IT. So I want to talk briefly about two recent studies, and perhaps I'll talk only about the first one in order to stay in time and have time for discussion, which addresses the question, do category selective regions exhibit categorical activation profiles across stimuli? The second one would be this one, and we can see if we get to that. So this was, as the previous study, done in collaboration with Marike Moore and also Douglas Ruff, Jerzy Bedoka, Peter de Weert, and Peter Bendettini, in whose lab I was a postdoc while we acquired these data. So the motivation for this is that we found these pattern clusters corresponding to faces, bodies, and inanimate objects. And we found these clusters for large ventral temporal ROI of object-responsive voxels, which included LOC, FFA, and PPA. So FFA and PPA are known to be category correlated, so they might explain this clustering to some extent potentially, right? However, when we removed them from this ROI or looked just at LOC by itself, the clustering was very, very similar, essentially the same at a sort of qualitative level. So here we want to have a look at what's going on in FFA and PPA, this clustering. So we could have a number of hypotheses. The first one is that there are overlapping pattern distributions when we look at FFA and PPA activation. And now this is an activation-based study, so we're looking at the average activation in those regions again, as in the classical approach. But the assumption that we're dropping is we're not predefining the categories, and we're looking at single image responses. So what is known is that faces activate FFA more and places activate PPA more, right? But without looking at single image responses, that leaves a lot of possibilities open, right? For example, you could have faces activating FFA and PPA, and places activating FFA and PPA, and some places or scenes might activate FFA more than some faces, right? So there could be overlap. So one possibility would be that these distributions are overlapping both multivariately and univariately. So in that case, we wouldn't get sort of category clustering for FFA and PPA, and that would mainly be due to the other regions in HIT, as would also be consistent with the finding that we didn't need these regions in order to get the category clustering. Another possibility is that we get these sort of skinnier distributions that are separate when we consider them multivariately. So we'd have multivariate separation, but we could still have univariate overlap. And finally, another possibility would be that FFA and PPA are sort of categorical independently of each other at the univariate level already, and that would imply that all the faces elicit more activity in FFA than all the places, and more generally all other object category members. So our specific questions were, is regional average activation greater for every preferred category object than for every non-preferred category object? And this could be called categorical rank ordering. Secondly, is the activation graded across the exemplars, or are there no significant differences? And the third question is whether there is a step-like drop of activation at the category boundary. Oops. So we're going to rank the stimuli by regional activation, and this is going to be the color code. So when we look at this for right FFA, here all the stimuli in the order in which they activated the right FFA. You see that the most highly activating stimuli are all faces, shown in red here. And then there are sort of a few bodies coming in between the faces, but not really any of the other categories. So it is it does look like quite a clear division. And this is strictly monotonically falling by definition, because we'd be showing the same data here that we've used to order the stimuli. So when we look at that for left FFA, it looks similar, but there is a little bit a bit fewer bodies in the sort of top group here. We can look at that with ROC curves, and we find sort of 0.82 for left FFA and 0.94 for right FFA, so quite a clear division. For PPA, the division is looks much clearer when you look at it like this. It's essentially perfect in both left and right PPA. And for early visual cortex and human IT, we see sort of all the categories mingled up together, even though there might be some sort of significant effects on average. So an important question is, when we look at this, we have these inversions here, right? So we have non-faces that drive FFA more strongly than some faces. And so we want to know, are these inverted pairs just due to noise in the data? Or in other words, are there replicable inverted pairs? Because that would indicate that it's not due to noise, but there's actual examples of non-faces that drive FFA more strongly than some faces. So we can address this by taking the ordering from session 1, then determining these inverted pairs, and then looking at what happens to these inverted pairs in session 2. So here the way I'm testing that, we did sort of several tests on this, but the latest one, and maybe the clearest one, is this one. What I'm showing you is the test set gap for training set inverted pairs. So we take the training set, which is session 1, take the inverted set, and then we look at the activation gap for these inverted pairs in the other data set. Does that make sense? So here I'm showing you this for right FFA defined at 128 voxels. And along the horizontal, I'm plotting the number of largest gap training set inverted pairs. And this is because this is sort of a slight statistical complication. So the easy thing to do would be to take just the maximum inverted pair, the one that has the largest gap in set data set 1, and then look in data set 2 whether that replicates. But since we have lots of stimuli, then we have very little data to assess this, right? So we want to average more. But the more we average, the more we might get into inverted pairs that are inverted just due to noise, right? So we want to do it for the whole range of possible set sizes of pairs. So if this is 0, then if this is above 0, then that would indicate a replication of the inverted pairs. And if it's below 0, it would indicate non-replication. And for the right FFA, what we see is this. So it's never significantly above 0. So that indicates that we have no evidence for any of the inverted pairs that we find in a given session replicating in the other session. So we can look at that for different regions. So this is what I've just shown you for right FFA. For left FFA, this looks very, very different. For left FFA, there seems to be very strong evidence of replicating inverted pairs. So there it's not nearly true, apparently, that every phase drives the region more strongly than every non-phase. For PPA, we see no evidence at all of replicating inversions. So as far as we know in PPA, for our stimulus set, it sort of perfectly orders them, as you know, at the given the amount of data that we have and the sensitivity that gives us. So the second question is the question of within category gradedness and categorical step. So the way we've looked at that here is by sorting the images, first by category, putting the preferred category first, and then by session 1 activation within the categories. So here for right FFA again, and we have all the faces here, and within the faces they're ordered by the activation, and within the non-faces they're also ordered by the activation. So when we do this for session 1, this is what it looks like. Monotonically falling in both of these categories by definition, right? And here there's a sort of steps up. This tells me this is my personal gong. Is that in line with yours? Oops. All right. That's nicer. Yeah, I agree. So here for session 1, we see this fall by definition because this is a session by which we've ordered the images within each of the categories. So we see this fall off within both categories. And the fact that this jumps up here is also not surprising because we're taking the largest one here. This is the first one within the non-preferred, right? So this step up might be entirely artifactual. And then we can look at session 2 data in this ordering and fit a simple model to it where we just fit a ramp, a line to the preferred category, and then a step function between the preferred and the non-preferred category, and then also a line within the non-preferred category. So our model has sort of a ramp for preferred, a ramp for non-preferred, a step, and a confound mean. That's all. It's a very simplistic model. And this now gives us some indication that's not biased by the ordering because we took the ordering hypothesis from independent data, right? And then we can cross-validate this and bootstrap across the image set. So we bootstrap resample the images with the motivation that we don't want our analysis to be heavily dependent on the particular images we chose. So we want to sort of simulate the variability that we would get if we had different stimulus sample from the same hypothetical distribution of stimuli. And this is what we get for the right FFA. We get a drop-off and a significant drop-off within the preferred category of faces. And we see a little step here, which is not significant, and a drop-off that's also significant within the non-preferred category. So this is the one that I've shown you. So let's place that in a little more context. This was right FFA. For right PPA, we get this very strong step here, which is highly significant. For early visual cortex, we get nothing significant at all. And we look in context across a wide range of ROI sizes. What sort of is most prominent is this clear and highly significant step for PPA, left and right, and sort of pretty consistent gradedness in the preferred and non-preferred for the right FFA. But in the left FFA is kind of a little bit less clear, especially for the faces. So our specific conclusions here were that regional, to the first question, whether there's a categorical rank ordering, the answer is yes for PPA and for right FFA. There we have no evidence for replicating inversions at all. For left FFA, there seem to be replicating inversions. For the second question, whether activation is graded across exemplars, the activation seemed to be graded for right FFA and to a lesser degree for the PPA. And is there a step-like drop of activation at the category boundary? For PPA, there's a very clear step, and for FFA, less so. So I think I'll stop here, leave the second part out, so we have some time for discussion.
Nico, it seems like these kind of conclusions are highly dependent on the stimulus set. Even though you're bootstrapping over stimuli, you're still limited by the set you started with, right? Yeah. So, thought experiment, if you just put in some very low contrast faces, maybe they give you less % signal change and your category boundary goes away, but there's still perceptually faces, right? So, and there's Roger Tutel's group had a paper of that sort recently, right? So I don't know, how do you, maybe this opens up a can of worms, but you're kind of pushing in, when you're going this analysis, you're sort of going with the idea that there is something that should be categorical, or I think that's what you're arguing here. Maybe I'm missing is that your argument, that these are really categorical areas? Or I don't know, this is really an open-ended question, so I think you see the issues, but we talked a lot about this at dinner when we were in, where were we? I don't remember, but so we're actively interested in this at the neuronal level, but so these are really cool analyses to see, but I wanted to get your thoughts on that now that you've done this.
I completely agree with that it's limited by the stimulus set, and if you could come up with something like this, like another explanation, then that might be a compelling explanation. So in that sense, it's never completely conclusive, right? And even though this bootstrap simulation is an attempt to simulate the variability, of course that is only for a population that these can be viewed as a random sample of. So in the example that you gave, that they're all lower contrast, then sort of, that's what you would get, obviously. So I completely agree with that. On the other hand, I think that is a problem with any study where we, no matter what analysis we do or what question we ask, we're always limited by our stimulus set, aren't we? So I think this is the induction problem that you're giving me, right? Isn't it?
Well, you could ground it in perception, right? So we heard that in some of the earlier talks, right? So my argument, if those low contrast faces, if you so low contrast that a human can't report them as faces anymore, then that's a sort of an objective definition of is it or is not a face, right? That is sort of there's ways to ground things that sort of release you from the stimulus set. There's sort of psychophysics, right? That would be one way to be objective about it.
Yeah, that's very, very useful to, yes, I agree with that. So the other question in the that I I heard was the question of what we should call categorical, right? I know that there's different opinions about the appropriate language, right? Whether, at what point is it appropriate to talk about categories at all. And to me, there is sort of no particular magic to something that I would call a categorical representation. So it wouldn't have to be perfectly categorical in some sense of, if you come up with examples, with untypical examples that clearly, fall on one or the other side of the line, and it didn't conform to that. And it also wouldn't have to do very high-level processing. So let's say if you had a region that processes the images just by template matching, but it's full of templates that look like faces, then I'd be very happy at sort of the higher level, the verbal level of describing the system, to interpret this as serving the function in the context of brain information processing of representing the category of faces, right? And that's sort of just a language convention to me that I don't think misleads us seriously.
So I like the replicability analysis, and I wonder if that could be applied to your original larger data set that includes everything as a way to try to discover some finer structure in that whole matrix of similarities, finer than just animate, inanimate, head and body.
So these are two questions, right? About the replicability of the representational dissimilarity structures and about finer divisions, right?
Well, I'm not questioning the binary divisions. Okay. I'm just saying within those divisions, is there repeatable finer structure in the similarity matrix, or is animate just like all mixed up and there's no meaningful nothing meaningful in that matrix, and maybe the replicability analysis that you've applied to just FFA could be applied there to
exactly. So I haven't talked about this part because it's published work and I've talked about it before. So it's we looked within the categories at the dissimilarity correlations between human and monkey, and also between human IT and different models, and between different brain regions as well. And basically, there are lots of significant correlators, lots of replicable structure within the categories. So for example, within the faces, within the bodies, within the inanimates, when you do split half analyses, you find replicable dissimilarity structure, and also between human and monkey. Right, so the human and monkey correlation of dissimilarities that we see is not driven just by the category divisions, but it's also significant within the categories, and also for the distances between the animates and inanimates even. So it suggests that for one thing, it sort of confirms that these are significant and that there's sort of continuous variation that's replicable. And on the other hand, it also shows that this continuous variation is somewhat consistent between the species. Mm-hmm.
So let me just follow that up, maybe. I'm really interested in the question of, so within that data set and within the FFA data set, where you show that you've got this replicable rank ordering of activations, what's the way forward for understanding what that structure correlates with, whether it's something semantic or structural, or is there a way forward? No.
Well, I think the way forward is to explore models at many different levels of abstraction, much like Jack is doing with like semantic level models, as well as models that process the images and do something with the images. And that would be sort of an empirical way also to come closer to Jim's question, I think, whether we can find a computational model, and if so, at what level of complexity that can explain the responses, right? So so another way to say this would be the semantic interpretation is kind of a placeholder. It doesn't really tell us what we want to know. It just summarizes the findings, right? It's just and I think it's not helpful to handcuff ourselves and always see it's all faces, but never say that because we want to remind ourselves that that doesn't mean that we understand it, right? So I think we should say, well, yeah, this seems to be responding to faces, but that doesn't mean we understand how it's computed. Now let's look at lots of computational models, try to explain it, and then we get this situation that we had, which is really the challenge to us, that the semantic model explains a lot of variance, right, and the computational models don't, and we have to close that gap.
And just a comment to follow up on that a little further. In FFA, the differences in activation, one model that you could test that's suggested by some results and some theories about how faces are encoded in a space where every neuron is representing distance in a particular direction from an average face. So an obvious thing that you could do with that FFA data is look at whether that rank ordering has anything to do with distance from average.
Yeah. And that's where specialized stimulus sets for particular hypotheses come in handy, right? I mean, this is another very interesting question. On the one hand, it's good to reduce the assumptions built into a design, but by using assumptions and choosing stimuli, of course, we can focus our sensitivity on questions of particular relevance like that one. And this would directly suggest using a face space and a face synthesis model to make a stimulus set that would optimally address that.
So on those lines, that's almost exactly the question I was going to ask, but I think maybe the easier way than dealing with the computational models of the stimuli, you have a really nice set of stimuli as they are now. Why not pare them down into sub-elements that exactly match them on certain feature dimensions? So the outline, to make it say unrecognizable as the object it is because you're missing texture markings or whatever, and remap the space, systematically eliminating subcomponents of the stimuli.
Oh, that's a very good idea as well, yeah.
Down what you have.
So, for example, go to grayscale, outline, different
stuff, and then be sure in some sort of psychophysical experiment that maybe people don't recognize them as the objects per se, and then sort out the visual component from the semantic component.
That's another interesting approach, yes.
Yeah.
Swaroop?
Swaroop, you go first.
So this is going back to the limitation of the stimulus set.
Yeah.
It's more of a question, like, suggestion from you. So if I have, say, all faces in your stimulus set, then I might actually get a good correlation between the model, like a V1 model or something, and FFA as well, like, say, higher level areas. So the correlation between the matrices doesn't really tell us how similar those two things are, unless there's a rich, meaningful structure in the matrices we're correlating. Is there a way to quantitatively know if the comparison matrices are good enough for the comparison?
Are you saying the correlation will depend on the stimulus set, and therefore it doesn't, in general, tell us the relationship between two representations?
What I'm saying is basically, if the matrices are not interesting enough, then even though they have high correlation between a model and a brain area, that doesn't necessarily tell us much. So is there a way to know if the matrices we are basing our analysis in the RSA on are interesting in some way? So
everything depends on the relevance of the stimulus set, I would say, right? So you could have, maybe Jack wants to comment on that. You could have a very unnatural stimulus set for which your model does very well explaining the responses, but then that tells you nothing about how it would do for more naturalistic stimuli. So to me, that is the the argument in favor of making stimuli naturalistic. And so we're on a continuum between experimental control and naturalism, where here we have Gabor's and here we have virtual reality interaction of a perfectly rendered environment. And there's sort of sweet points along this whole continuum, right? And we want to choose a point, right? So movies are a step up from, I think, static stimuli, and we need to think about how to get stimulus samples that are somewhat representative of real-world experience. So here we've just sort of tried to span a wide range of categories, but of course it's very limited, right? And also biased by the frequencies, obviously. So it's a difficult problem. It's at the interaction between our natural image statistics and natural world statistics, really, and studying what the brain does.
I'm going to take the last question here, and that is the semantic model that you're using is basically the animate-inanimate faces versus bodies. Have you tried to do a more detailed semantic model like in Jack's work, where you have a list of descriptors, and each descriptor is a dimension, and so you try to characterize the dissimilarity in a larger, higher dimensional semantic space?
Yeah. We haven't really gone very far with that. What we did do was sort of finer category distinctions, and they're also correlated, but they don't explain as much variance. But they're significant. So if you add more distinctions to a sort of composite model of the dissimilarities, then these are definitely correlated. But what we have yet to do is get sort of continuous semantic vectors across the stimuli and then see what they explain.